Okay, hi everyone, welcome to the session. So this is lecture 4 of MD 6028. What we're going to talk about today is genome replication. So last week we talked about genome expression, this week specifically focusing on genome replication. As you'll see, the two processes are really tightly linked, but it makes sense to look at them one by one. So again, we've done genome expression, now we're moving on to genome replication. And similarly to last week, we're going to work through the groups of viruses one by one. And we're going to look at how members of each of these groups engage in the process, and hopefully everything will become clear. First off, we're going to establish some key principles of genome replication. We're going to talk about what we mean by genome replication, and we're going to look at some definitions and things like that. So this slide outlines the process of host genome expression that we were looking at last week. And I'm putting this up so we can contrast it to host genome replication, which we're looking at this week. So with host genome expression, we have a DNA genome, and then this DNA genome is being used to produce RNA. This process is transcription. Then this RNA is translated into protein. And this is one genomic process that occurs in the host cell. It's genome expression. The other genome process is host genome replication. So this is a second genomic process that is occurring in the host cell. And this is where the genome is being copied, essentially. That's why we call it replication. And as you can see, the endpoint of host genome replication is an exact copy of the original genome. So this copy is in the same format, it's double-stranded DNA. Now we say exact copy knowing that that isn't quite always the case because we have mutations, but apart from these mutations, we can consider the product of genome replication as being an exact copy of the original genome. The sequence is the same, the composition is the same. Again, it's double-stranded DNA. And this is one of the key principles at the center of genome replication. The principle is that the end point should be the same as the start. So the product should be essentially exactly the same as the original genome. If it's not the same, or if there's significant bits missing, then that's a massive problem because you get this very rapid loss of genome function if you're losing chunks of the genome, or if the genome goes from DNA to RNA. It doesn't make sense. It's not genome replication. So genome replication involves a near exact copy being produced. And again, these are the two principal genomic processes that are occurring in a cell. We want to understand these processes in detail and look at the enzymes that are mediating the processes. And for genome expression, we're concerned with RNA polymerase because this is the enzyme that's producing the RNA. We also have the ribosome, which is producing the protein from the RNA, but we're really considering this process of transcription because if we remember, transcription is what's different sometimes in host cell genome expression compared to viral genome expression. So we really care about the enzyme here. In the host cell, as we've discussed, the enzyme that mediates this process is a DNA-dependent RNA polymerase. It's an RNA polymerase because it's producing RNA, and it's DNA-dependent because it's using the DNA genome as a template. Again, DNA-dependent RNA polymerase. Now for host genome replication, the same principle applies, but it's just a different enzyme because it's a different process. Okay, but the naming of the enzyme is completely consistent with what we've talked about. So the enzyme that mediates this process is a DNA-dependent DNA polymerase. It's a DNA polymerase because it's producing DNA, it's polymerizing deoxyribonucleotides, and it's DNA dependent because it's using the host cell DNA genome as a template. So clearly it's a DNA dependent DNA polymerase that is facilitating host genome replication. And again, reiterating this key point here that when we're talking about genome replication, we're talking about a process that produces a copy of the original genome. So it's a copy that's identical or near identical because we can't ignore the fact that submutations can occur. Again, this means if the genome is DNA, which it is in host cells, then the end product must also be DNA because it must be identical. when we're thinking about viral genome replication, which is what we will think about today, the genome again needs to be copied exactly. So if the viral genome is a positive sense RNA, then the end product needs to be positive sense RNA, because the end product needs to be identical. And this is identical not just in molecular format in the sense that it is RNA, but also in the sense that it is positive RNA, because it should be exactly the same. The strand sequence should be exactly the same. So if the original genome is positive sense, the genome copy should be positive sense. Same for negative sense, and same for any other genome format. It needs to be an exact copy. Okay. So now what we want to move on to and talk about is, okay, so what we're going to do today is look at this process in the different groups of viruses. So we're going to work through the different Baltimore groups of viruses. So we're using the Baltimore classification system, and we're going to look at how these different groups engage with this particular process of genome replication. First off, we're going to talk about DNA viruses. Now, in the last lecture, we looked at how DNA viruses engage in genome expression in a very similar way to host cells. And the reason for that similarity is because they have the same genome format, right? They have the same genome composition. It's DNA. Some respects it's similar in DNA virus genome replication. So in some respects there are key similarities between the host process and the viral process, but the degree of similarity is different for different DNA viruses. So some DNA viruses will rely very heavily upon host cell proteins. Some DNA viruses will rely less heavily upon host cell proteins. But we can understand that because DNA viruses have a DNA genome, which is the same genome as host cells have, right? It's DNA genome. All DNA viruses could, in theory, just use the host genome replication machinery. So they could, in theory, just use the DNA-dependent DNA polymerase that is present in host cells. However, that's not necessarily what we always see. So some viruses, even though they have a DNA genome, have evolved an extensive ability to replicate their own genome essentially, and they still can only do this inside a host cell because, and here's the key issue, all DNA viruses require some host components to replicate their genome. So as we know, viruses are always dependent upon the host cell, and this is still true for DNA viruses. However, the level of dependence differs across different DNA viruses. So some are really tightly reliant upon the host cell. They essentially use almost all the same machinery and enzymes, but some DNA viruses are less dependent, and we're going to look at some examples now. So again, just this key phrase, in principle, DNA viruses are able to directly co-opt host genome expression machinery, which we've talked about, and genome replication machinery. However, despite this, DNA viruses will encode some of the components or machinery involved. So particularly for DNA genome replication, these viruses will encode at least some of the machinery that's required to engage with this process. And we can generally split DNA viruses into two groups, and by splitting them into two groups, we can say some DNA viruses are heavily reliant upon the host and some have more independence, though they are fundamentally still dependent upon the host. They're just less dependent upon the host, we can say. And again, now we're talking about genome replication, so we're talking about in the context of genome replication. We can label these two groups of DNA viruses as being the small DNA viruses and the other group being the large DNA viruses. The small DNA viruses encode minimal elements of the replication system. So these small DNA viruses are heavily reliant upon host machinery to replicate their genome. The large DNA viruses, on the other hand, these actually encode most of the replication system themselves. However, as we've said, even with these large DNA viruses, they are still dependent upon the host enzymes and machinery to a degree. So all viruses are dependent upon host cells ultimately for these processes to be complete. As we've said plenty of times, viruses are obligate parasites. They need the host cell for these processes to complete successfully. Therefore, even though the large DNA viruses carry a lot of the machinery themselves, they still require the host cell for at least some components. Okay, so let's look at some examples. So a large DNA virus, we can think about the adenoviruses, so member of the Adenoviridae viral family, double-stranded DNA viruses. They encode the main part of the machinery themselves, and the main part of the machinery for genome replication is that polymerase that mediates the process. So as we've said, when we're going from DNA to DNA, we need a DNA-dependent DNA polymerase. In the case of these large DNA viruses, they encode their own. So adenoviruses carry their own DNA-dependent DNA polymerase, but there are other accessory factors, associated factors that they need and that they can only use host cell versions of. So they rely upon the host cell for things like transcription factors and for topoisomerase as well. And you'll know what these are from kind of biochemistry and cell biology lectures and things like that. And the key point here is even though they have their own, even though they carry the main enzyme themselves, even though they have their own DNA-dependent DNA polymerase, they still rely upon the host cell for some aspect of genome replication. On the other hand, we have the small DNA viruses, and the example here we have is Polyomaviridae, so members of the virus family Polyomaviridae. We can call them the polyoma viruses. These are a type of small virus. They do not encode their own key enzyme. They do not encode their own DNA-dependent DNA polymerase. This isn't a problem in terms of the virus engaging in genome replication, because as we've said, these are DNA viruses, so their genome is in the same format as the host cell genome, and therefore they can just use the host cell DNA-dependent DNA polymerase, because this is the enzyme that is facilitating host genome replication as well. However, even though they rely upon this enzyme, they still encode some elements themselves. So they still encode an ATP-dependent helicase. And therefore we have this kind of balance. So all of these different DNA viruses, they ultimately rely upon the host cell for at least something, but they also carry some components of it themselves, if that makes sense. And the small DNA viruses are more dependent upon the host because the majority of the components are coming from the host. But for the large DNA viruses, the majority of the components they're bringing in, crucially this big DNA-dependent DNA polymerase enzyme, which is the key enzyme. However, they're still reliant upon the host for other factors, for associated factors that enable the process of genome replication to occur. So every single different one of these DNA viruses will be bringing some factors in themselves and will be using some host factors also. Okay, and those are the principles we need to understand for DNA virus genome replication. Again, there's a little bit of complexity there. The key takeaway is the points that we've really defined quite clearly, so that these viruses could, in theory, all use host DNA-dependent DNA polymerase because their genome formats are the same. But in practice, there's a little bit more complexity there, and we have to understand the difference between small DNA viruses and large DNA viruses. Okay, so what we want to talk about now are the single-stranded RNA viruses. So we have the positive sense single-stranded RNA viruses and the negative sense single-stranded RNA viruses. Fully confident you understand the distinctions between the two by now. And again, we're therefore going to jump straight in and look at the differences in genome replication between these groups. Okay, so bringing this back to the Baltimore classification system, we're talking about group 4 and group 5. So the positive sense single-stranded RNA viruses in group 4, then the negative sense single-stranded RNA viruses in group 5. And first, we'll focus on the positive sense single-stranded RNA viruses. So these are class 4. Okay, so here we have a version of essentially genome expression for the positive sense single-stranded RNA viruses. So if you can remember, this is a simplified version, but if you refer back to last lecture, it should make sense. It's just a little bit less information there, but the key points are made. There are essentially two routes that the initial positive sense genome can take once it enters into the host cell. So it can be directly translated into protein, or we can see it being used to produce a negative sense complementary strand. And then this negative sense complementary strand can then be used to produce a positive sense complementary strand, because again, this is how these polymerases work. They progress along the existing strand and make a complementary strand of the opposite polarity. So if you have a strand that's single-stranded with positive polarity, an RNA polymerase functions to make a copy of it, it's actually going to make a complementary copy of the opposite polarity. So we're going from this initial positive strand to a negative strand, and then another positive strand is being made from this negative strand, and this positive strand can then be translated into protein, because as we've discussed at length, these positive strands function exactly the same as cellular mRNA. They can be translated into protein because they're positive, so they contain the order that will determine the order of amino acids in the protein. So for some positive sense single-stranded RNA viruses, genome replication is essentially the same as genome expression. Okay, and we can see that from the diagram here, because we can see that by the process we've just described, which is core to genome expression, the end product is the same as the start product. So essentially genome replication has occurred. That's what genome replication is. Genome replication, as we've said, is the creation of a product that is exactly the same as the initial genome. And that's what we're seeing here. We're seeing a positive sense genome that is being processed, and then ultimately a positive sense genome copy is made. Now there's this middle step here that involves the production of negative sense single-stranded RNA, and this is just an intrinsic part of the genome replication of these viruses. Again, like genome expression, it involves this intermediate, so it involves this step. So we can say for genome expression of many positive sense single-stranded RNAs, the initial positive sense single-stranded RNA genome is used to produce a complementary negative sense copy. This negative sense strand is then used to produce a complementary positive strand copy, and this positive strand copy should be exactly the same, give or take mutations, as the initial viral genome, and therefore this copy can serve as a new viral genome for the progeny that are being produced. So it is a successful product of genomic replication. And this is what that looks like. So our example here is poliovirus, and this is how poliovirus, being a positive sense single-stranded RNA that replicates in this way, this is how polioviruses replicate. So initially, the positive sense single-stranded RNA genome is used to produce a negative sense single-stranded RNA copy using RNA-dependent RNA polymerase. The same enzyme then facilitates the production of positive single-stranded RNA. And then this positive single-stranded RNA copy is packaged into new virus particles, because again, it is a copy of the viral genome, so it can function as a viral genome. And this virus particle with this copy inside is potentially infectious, so it can leave the host and infect new cells or infect a new host. So quite a straightforward process if we understand the process of genome expression that we talked about last week. The only real difference is that instead of the positive sense single-stranded RNA being translated into protein, it's being packaged into new virus particles. And again, there's some specific examples of virus families that engage in this process here on the left. And as you'll see, these are all positive sense single-stranded RNA viruses. So they're class 4 in the Baltimore classification system. And what we can see on the slide is the phenotypic classification system. And you can see they all fall under the single-stranded positive sense arm of the RNA virus map. Okay, and some of these viruses are significantly important. So poliovirus, like we said, West Nile virus, Zika virus, these are members of Flaviviridae family, which is a virus family that contains many members of significant human disease. So we're talking about quite important viruses here. So it's yeah, yet another reason for us to understand this process. Okay. In reality, the process can get a little bit more complicated than that. So the key principles are what we've just talked about, but there's a little bit more as well. There's a little bit more that we need to understand for many viruses. Again, we've looked at some where that's exactly what happens and that's fine, but there's also more to the story for some positive sense single-stranded RNA viruses, and it's important we understand that. So that's what we're going to look at now. Okay, and on this slide, we can see genome expression is demonstrated for positive sense single-stranded RNA viruses. Now, this is something we looked at in detail last week. However, you'll see one of the stages is a little bit different. So you'll see in kind of the third part of the stage, you will see these what we call subgenomic fragments. So you will see portions of the genome. You will see portions of positive sense single-stranded RNA. And again, these are subgenomic fragments. And for many positive sense single-stranded RNA viruses, this is how genome expression actually occurs. So let's look at this in a little bit more detail. So normally, or we think, well, not normally, but what we looked at last week was this standard way in which many positive sense single-stranded RNA viruses express their genome. So they start off with a positive sense strand, produce a negative sense strand from that, and then produce a complementary positive sense strand to that negative sense strand, and this positive sense strand is then translated into protein. And for many positive sense single-stranded RNA viruses, this is exactly what happens. However, for some, the positive sense strand that is produced from the negative sense strand, so this later stage in the process, if you like, so it starts off positive strand, then goes negative strand, and then another positive sense strand is made. However, for many of these viruses, this final positive strand is actually produced as subgenomic fragments. So it's produced in portions, and these portions are then translated into protein. We could ask why this happens, and the answer is because it gives the virus a little bit more control about how the genome is expressed. So it is often the case that as more and more production happens, so more and more genome expression happens, the size of these subgenomic fragments changes. And there's specific mechanisms that viruses use to initiate this change. And what this change essentially means is that viruses can start off producing particular size subgenomic fragments, but as genome expression goes on, they can make bigger fragments. Okay, and ultimately what is occurring is the smaller fragments are translated into proteins that are important for the early stage of infection, and the larger fragments that are produced later are translated into proteins that are useful for the later stages of infection. So these subgenomic fragments are utilized in genome expression for many viruses. And again, the purpose of this is it enables the virus to have more control over what proteins are being made when. So again, a little bit more complex than we looked at last week, but the principle is exactly the same. The overall stages are the same. The only additional complexity is the idea that rather than a full transcript being made from the negative sense intermediate, we have subgenomic fragments being made from the negative sense intermediate. The problem, however, with this system is that the production of these subgenomic fragments is not sufficient for genome replication. So as we've said, for genome replication, the overall process is the same in terms of stages as the stages of genome expression for these positive sense single-stranded RNA viruses. So they start off with a positive sense strand, then they produce a negative sense complementary strand, then they produce a positive sense strand that is complementary to that negative sense intermediate. And we've said this positive sense strand can be translated into protein, or it can be packaged into the virus particle. And this is what happens for poliovirus and some other positive sense single-stranded RNAs, and that's absolutely fine. However, again, there's a problem with this diagram we're looking at now, and the problem is many positive sense single-stranded RNA viruses, as we can see, express their genome through subgenomic fragments. So they therefore can't just package these subgenomic fragments into the viral genome, because it's not into a new virus capsid, into a new virus particle, because these subgenomic fragments are not full copies of the genome, they're just partial copies of the genome. So for these particular positive sense single-stranded RNA viruses, genome replication can't be exactly the same as genome expression, because the product of genome expression is not a complete copy of the genome. And as we've said, the end product of genome replication needs to be exactly the same as the initial genome that is being copied. Therefore, these particular positive sense single-stranded RNA viruses need a mechanism to switch from genome expression. So they need to stop producing subgenomic fragments from the negative sense intermediate and start producing full-length genome copies from the negative sense intermediate. So they need to go from this process on the right, genome expression involving subgenomic fragments, to this process on the left, genome replication, so producing a full-length copy of the viral genome, this full-length negative sense single-stranded RNA copy. And these full-length copies are essentially identical to the initial genome. Again, putting aside mutations, they're essentially identical, and therefore they can be packaged into the virus particles. So hopefully this makes it clear. Again, we're just adhering to these rules of genome replication that we've talked about. So again, as we've said, there needs to be a switch. So these particular single-stranded RNA viruses are using subgenomic fragments for genome expression. Therefore, genome replication has to be different. They have to switch from producing these subgenomic fragments to start producing full-length genome copies. So how do they actually control this switch? It's quite a complicated fact. Again, it's a specific process that we're going to look at now. It's different for different viruses, but often the key principles are similar or analogous at least. So it's going to help us to look at some examples, and that's what we're going to do now. So we're going to look at Synbis virus, which is a member of the Togaviridae family. Again, it's a single-stranded positive sense RNA virus. Like all the ones we've looked at in this section of the lecture. So what we want to consider now, as we said, is how does this Synbis virus, which is a positive sense single-stranded RNA virus that expresses its genome using subgenomic fragments, how does it switch from these subgenomic fragments to producing full-length genome copies for genome replication? So how does it switch from genome expression to genome replication? Okay, so this is a diagram of specifically what is occurring when Synbis virus is expressing its genome. Obviously, it's a positive sense single-stranded RNA virus, as we said, so it has a positive sense single-stranded RNA genome. This is used to produce a negative sense complementary single-stranded RNA copy. And then in genome expression, we then have subgenomic fragments of this negative sense single-stranded RNA strand being produced. And as we can see here, it's really this key 26S subgenomic mRNA fragment. So it's this one fragment. However, the issue is this one fragment is subgenomic. It's smaller than the actual full-length genome of the virus, so it can't be packaged into a virus particle because so much information is missing. It's basically just like packaging in half the genome. big problem. So what the virus essentially needs to do in this situation is switch from the production of the subgenomic fragment to the production of full-length genome copies. Okay, so we want to understand this switch from genome expression to genome replication, from the production of subgenomic fragments to the production of full-length copies. So the processes that we're looking at in the slide are the processes of RNA being produced from RNA. Okay, so we talked about this last week and the idea that to mediate this process, an RNA-dependent RNA polymerase is needed. Now, the particular RNA-dependent RNA polymerase that facilitates the process in Synbis virus is something called P1234. So this is an enzyme that is encoded in the viral genome. So the positive sense single-stranded RNA genome of this virus is translated as soon as it comes into the cell, and it's translated into this protein. So P1234 is something that's produced. This P1234 is, as we said, the RNA-dependent RNA polymerase that facilitates the production of RNA copies of the viral genome. So initially you have the positive strand, it's translated into protein. The protein that is made is P1234. Then this can then produce a negative sense copy of the positive sense viral genome. And then this negative sense copy is used to produce 26S subgenomic mRNA, so a subgenomic fragment. Another protein is also made by translation of the viral genome, and this protein is called NSP. NSP, as this process is going on and on, will accumulate in the cell. So as you get more and more expression of the viral genome, so more and more translation, there'll be higher levels of NSP accumulating in the cell. And what NSP essentially does is it has an impact on P1234. It processes it, we say. So it changes the enzyme in such a way that the enzyme's function changes slightly. And this change in function ultimately results in a different length transcript being produced from the negative sense genome intermediate. This transcript is referred to as 49S genomic mRNA. It's referred to as genomic mRNA because it's not subgenomic, it's not a fragment, it's actually a full-length copy of the original genome, and therefore it is a functional product of genome replication. It can be packaged into new virus particles and can function as a genome in and of itself in the new virus particle. So what we have here is a full switch. So we've gone from the production of subgenomic mRNA to the production of genomic mRNA. So again, a switch from genome expression to genome replication. Okay, so to summarize, we can say we have two principal methods of genome replication for the positive sense single-stranded RNA viruses. The first one is very straightforward. It's just the idea that full positive sense transcripts identical to those used for genome expression can be packaged into the virus particles. That's what we looked at with poliovirus. The second strategy is that there needs to be an active switch between the production of subgenomic fragments for genome expression and the production of full-length genome copies for genome replication. Okay, so that's the positive sense single-stranded RNA viruses. Again, there is complex information there, there is technical information, but it's all adhering to the principles we've established. So if we understand the principles that we've established in earlier lectures and at the start of this lectures, then there shouldn't be anything too difficult for us to understand there. Okay, so next we want to think about the negative sense single-stranded RNA viruses and how they replicate their genomes. Okay, so we're talking about negative sense single-stranded RNA viruses again. So we're talking about class 5 on the Baltimore classification system, and we're talking about this arm of the phenotypic system. So RNA viruses, negative sense single-stranded. Okay, So for negative sense single-stranded RNA viruses, we have to introduce the same extra bit of complexity that we've just introduced for positive sense single-stranded RNA viruses. So this idea that genome expression doesn't just necessarily involve full-length positive strand copies being made, but actually involves subgenomic fragments being made. So again, as we can see here, this is the case for negative sense single-stranded RNA viruses in genome expression. Positive sense RNA is created from the negative sense genome, but this positive sense RNA is made in subgenomic fragments, so in these fragments that are then translated into protein. Again, this creates something of a barrier to genome replication because genome expression and genome replication can't be exactly the same processes here, because genome expression involves the production of subgenomic fragments, whilst genome replication requires the production of full-length copies of the genome. Again, genome replication must produce exact copies of the original genome, give or take mutations. So again, there needs to be a switch from genome expression to genome replication. There needs to be a switch from the production of subgenomic fragments to the production of full-length genome copies. Because we're talking about negative sense single-stranded RNA, there also needs to be another step, because the production of a full-length positive sense single-stranded RNA is not sufficient for genome replication because it's in the wrong format, because it's positive sense and the original genome is negative sense. So this additional final step is needed for this to make sense, and this final step is the production of a negative sense genome copy from the positive sense transcript. So again, we're seeing something quite similar to what we see with many positive sense single-stranded RNA viruses, but we also have this additional step on the end. So now we again want to think about how this switch is controlled. So how do these viruses actually go from producing subgenomic transcripts, so genome expression, to producing full-length genome copies for replication? To explore this idea, we'll look at an example like we did for the positive sense single-stranded RNA viruses, and the example we'll use is influenza virus. As we can see from this phenotypic graph here, or this phenotypic tree, the RNA viruses are demonstrated here, and influenza virus is part of the Orthomoxiviridae family, and this family is a single-stranded negative sense RNA virus, meaning it has a negative sense single-stranded RNA genome. So let's have a look at how this virus switches from genome expression to genome replication. It expresses its genome by the mechanism we've just looked at, so the production of subgenomic positive RNA fragments from the genome, and then these subgenomic fragments are translated into protein. Now, one of the proteins that is made is referred to as NP protein. So this protein is directly made from the translation of the subgenomic fragments. As this process of genome expression continues and continues, the cellular levels of NP protein increase. It accumulates as it's being made more and more and more. And then again, what happens is the concentration of NP protein increases within the cell. When the concentration of NP protein increases to a critical point, it begins to coat the new strands of RNA, so the new transcripts, as they are being produced from the negative RNA genome. So the negative RNA genome is being used to produce subgenomic fragments by RNA polymerase. However, as NP proteins increase within the cell, NP protein binds to the transcript as they're being made and prevents them being processed into subgenomic fragments. So it actually maintains the integrity of the nascent strand of nucleic acid, nascent meaning newly forming. And as a result of this NP binding, the subgenomic fragments are never formed, but what is formed instead is a full-length genome copy. So again, this NP binding is essentially maintaining the integrity of the nascent transcript. It's ensuring it stays one full-length transcript, because again, this is what is needed for genome replication. So the accumulation of NP protein within the cell itself triggers the switch from genome expression to genome replication. So this enables the virus to express proteins when it comes into the cell, so produce these subgenomic fragments and produce protein as a result of translation, but then to move towards genome replication in the later stages of infection. So we know there has to be another stage as well, because this positive sense single-stranded RNA, which is full length, then needs to be used to produce a negative sense complementary copy, because again, we're talking about a negative sense single-stranded RNA virus, and the end product of replication needs to be the same as the initial genome. And this is what happens at the end. So again, let's just remember this final step, because it's really easy to miss it out, and then all of a sudden nothing makes sense because we haven't actually ended up with the type of genome that is necessary for successful genome replication. Okay, now let's look at another example. We'll look at vesicular stomatitis virus, VSV, which is a member of the Rhabdoviridae virus family, single-stranded negative sense RNA virus. And again, the virus is using these subgenomic fragments for genome expression, and it needs to switch to the full-length transcript production for genome replication. And the process is the same. It's NP protein again, so vesicular stomatitis, NP protein, which is going to be binding to the nascent transcripts. And by binding to the nascent transcript, it's preventing subgenomic fragments being formed and is maintaining the full-length transcripts, which can then be used as templates to produce the full-length negative sense copies, which again is what genome replication is in the case of these viruses, because they are negative sense viruses. Okay, so those are the single-stranded RNA viruses. Again, some really useful examples there. Again, hopefully you can see we're just applying these concepts we've already learned. So there shouldn't be anything too difficult there. If anything is giving you any trouble, just go through it a few more times. Just make sure you've listened to how I've explained it. Make sure you've looked through the slides yourselves. And as always, if you're struggling despite doing that, you have the course textbook which goes through these concepts very clearly. Okay, so next we want to think about the double-stranded RNA viruses. So again, working through these Baltimore groups. Next, double-stranded RNA viruses. So how do these viruses replicate their genome? Okay, so when we think about the double-stranded RNA viral genome, we can think of it in the same way as we think about double-stranded DNA. So it's these two strands that are paired, and one is a positive strand, one is a negative strand. So we have this pairing that occurs, and they're complementary to each other. If we want to think about what enzyme is going to be involved in genome replication, it has to be an RNA-dependent RNA polymerase, doesn't it? Because we're starting with an RNA genome and we're ending with an RNA genome, so therefore the enzyme that's involved in that process must be an RNA-dependent RNA polymerase. And what we're going to consider is how members of the family Virolidae replicate their genomes, because these family members have double-stranded RNA genomes, which is what we're talking about here. Okay, so we have an example virus here. This is a reovirus, or member of the Virolidae family, so reovirus. And what we're going to do is consider how this genome is replicated. As we can see, it's unenveloped, just so this diagram makes sense. So double-stranded RNA genome in the middle, then the protein shell on the outside. Okay, so initially positive RNA copies are produced within the virus particle, then they're essentially exported out into the cytoplasm. So an RNA-dependent RNA polymerase is working on the genome and producing positive sense RNA copies. These positive sense copies are exported into the genome, where they are translated into protein. This protein is then able to assemble into new virus particles. So again, the protein capsid is just a protein, sorry, the viral capsid is just a protein shell. That's why we call it a protein capsid sometimes, because it's a capsid composed of protein. So these proteins that are being made, viral proteins that can form together to form a capsid. As we know, each virus particle that's made needs a copy of the viral genome, and the way it gets the copy, or at least the positive strand of the copy, and that's important because obviously it's only the positive strand that is there at this point, is it is just incorporated into the protein shell. So as the protein shell assembles, the positive strand that has been made by RNA polymerase is incorporated into the virus particle. At this point, obviously, there's something missing, which is the complementary negative strand that pairs to the positive strand. Because as we know, as we've said many times, the end point of genome replication has to be the same as the start point. And these are double-stranded RNA viruses, so we know the start point is double-stranded RNA, so the end point can't be just a strand of positive sense RNA. It has to be double-stranded RNA. So the last stage explains this, and it is where the positive strand of RNA is used to produce a complementary negative strand of RNA, thus completing the double-stranded RNA structure. So let's also briefly consider the enzymatic processes that are occurring here. At each stage, and there's two different enzymatic processes really, but at each of these two stages, the process is going from an RNA template to a new RNA strand. So therefore, it must be RNA dependent RNA polymerase facilitating each of these steps. So initially, the double-stranded RNA genome is used to produce negative sense single-stranded RNA, RNA to RNA, so it's RNA dependent RNA polymerase. And in the final stage, the single strand of RNA is turned into a double strand of RNA. So again, this is RNA dependent RNA polymerase because it is producing RNA from an RNA template. Okay, and those are the class 3 viruses, so it's double-stranded RNA. And what we want to do now is think about class 6 and class 7. So class 6 and class 7, these are the unusual groups, should we say. These are the groups that engage in reverse transcription. And again, it's difficult to understand these groups looking at the phenotypic system because they sit within another classification. So group 7 looks like it's just a normal enveloped double-stranded DNA virus. It sits with the others, but based upon the Baltimore classification system, we know there's something different going on there. And same for retroviruses. So on the phenotypic system, we can't really distinguish the family Retroviridae from other enveloped single-stranded RNA viruses with a positive sense genome. But on the Baltimore classification system, we can differentiate this family. And the reason is because, like the class 7 families, it engages in reverse transcription. Again, we're not going to go into that in detail now, but we are going to go into it at some point. So we just wanted to point out that they are separate from the other groups, and we'll explore specifically how they engage in these processes at a later point. Okay, finally, so we want to jump into genome structures and actually look at what these genomes we're talking about appear as in terms of structure. So it's quite simplistic for us to consider that if we have a particular genome, it just exists as a kind of chromosome-like structure, which is what we sometimes think of when we're thinking about genomic material. It's a little bit different for viruses, and it's also not always the case that it is just a single structure within the virus particle. So we're going to actually look what these genomes can sometimes look like. When we have Reoviridae, we can have what the structure we see on the right here, where the genome is actually segmented. So it's still a double-stranded RNA genome, but there's multiple segments of it. So we have this large initial segment, then a smaller one, then a smaller one. Okay, and all the genetic information is there. and all the information that is there when the virus enters the cell is there when a new virus particle leaves. So these aren't subgenomic fragments. These are just segmented genomes. This is just where the entire genome is split up into constituent pieces. If you think about influenza virus as well, this has a segmented genome. So influenza virus has its genome split onto like essentially circles, so they're not quite circles, they're just linked ends. So that you can just think of them as linear genomes that are linked at the end, and they have multiple circles, so multiple segments within the virus particle. And some of these viruses will have genomes shaped like this. HIV, on the other hand, doesn't, so it has a non-segmented genome, but it does have 2 genome copies in the virus particle, so that's another type of variation we can see. Not every virus will have just 1 copy of its genome in the virus particle, some will have 2. We may know that influenza virus is known for being quite varied. So there's a lot of different variants of influenza virus that exist, and one of the reasons is because of these segments. So there can be almost chopping and changing in terms of which versions of which segments go into a new virus particle. And one thing that this variation can result in changes to the hemagglutinin and neuraminidase on the virus particle surface, and this is why we have all the different types of influenza, which we define by their H number and N number. What we actually mean by that is what's the hemagglutinin number and what's the neuraminidase. So what type of hemagglutinin do they have? What type of neuraminidase do they have? This impacts a lot about the viral replication cycle, and the variations we see in terms of hemagglutinin and neuraminidase are quite tightly linked to this particular segmented structure that the viral genome has. So here are some different DNA genome structures that we can see in viruses. So sometimes it will just be linear. So for poxviridae, so the pox viruses, there is linear double-stranded DNA genome. However, sometimes with double-stranded genomes, they'll circularize, so you'll get a circle of double-stranded DNA. And if there's single-stranded DNA, these typically exist as linear strands. And again, if we remember from our last lecture, these linear strands of DNA are usually turned to of single-stranded DNA usually turn to double strands very quickly anyway. Okay, we can have gapped genomes in the virus capsid as well. So some viruses will have gapped genomes. Again, I think this is something we mentioned. It essentially means there is a circularized double-stranded DNA genome that isn't completely double-stranded DNA. So there's a portion of the genome where there is only a single strand. Now again, this can be potentially repaired into double-stranded DNA, can't it? But it naturally exists as a gapped form, so there is this gap here. Okay, well that's everything we wanted to talk about today. We needed to talk about today, I should say. I look forward to meeting you again in the tutorial and going through the questions. Feel free to turn up with any questions you have, or ready to ask me to explain anything again that wasn't quite clear. Okay, thank you.